Hello, everybody. Welcome to the new lecture of our co-ecologies semester and program here at Fabrica. We are really happy to organize this lecture together with the Biodesign Challenge. We are happy to have today with us its founder, Daniel Grushkin, and Dr. Elizabeth Hénaff, who will follow up with a lecture. We are very happy that we can introduce both's work, and also very happy that also our Fabrica residents will have this opportunity of understanding the reality of Biodesign Challenge, and also have the opportunity of witness this lecture with the hopes of a long-lasting collaborations with them. Just to have a little bit of a short introduction, I'm going to read a bit of the biography of both, so they don't have to go through that. I'm happy to introduce Daniel Grushkin, who is the founder of Biodesign Challenge, an international student competition and education program that partners artists, designers, and scientists with students to imagine and create the future of biotechnology. He's also the editor of the Biodesign anthology Grow the Future, and also the co-founder of Genspace, a nonprofit community laboratory dedicated to promoting citizen science and access to biotechnology. He was the fellow at Data and Society and at the Woodrow Wilson International Center for Scholars, and an emerging leader in biosecurity at the Johns Hopkins Center of Health Security. As a former journalist, he has reported on the intersection of biotechnology, culture, and business for publications including the Bloomberg Businessweek, Fast Company, Scientific American, and Popular Sciences. He's currently a visiting scholar at the American Academy in Rome. And also we have with us Dr. Elizabeth Hénaff, who's a computational biologist and designer. She's currently assistant professor at the Technology, Culture, and Society Department at the New York University, the Tandon School of Engineering. At the center of her research is a fascination with the way living beings interact with their environment. Her inquiry has produced a body of work that ranges from scientific articles in peer-reviewed journals to collaborations with landscape architects to artist residencies in environments from the SVA to the MIT Media Lab. And her academic trajectory started with a bachelor's in computer science, followed up by a master's in plant biology in UT Austin, and a PhD in bioinformatics from the University of Barcelona, and also a postdoctoral work at Weill Cornell Medicine and the MIT Media Lab. So without further ado, I really want to welcome Daniel and Elizabeth for this fantastic lecture. Thank you very much for doing this, and thank you very much for your time.
I just want to thank you, Carlos, and I want to thank the entire Fabrica team for doing this with us. This is a really special occasion for us, and I think we're gonna have a good time. So let's just jump in and get started. So here's my opening slide. Carlos did a great job in introing me, so I'm gonna skip some of those initial slides. But my name is Dan Grushkin. I'm the founder of two nonprofits. The first is called Genspace. It's a community biology lab. So it's a space where anyone from any background can come into the laboratory, learn the fundamentals of molecular biology and biotechnology, perhaps bioengineer their first organism, look under the microscope, see their first microscopic world, and then go from there. And the second nonprofit that I founded is Biodesign Challenge. And Biodesign Challenge is a student competition where we partner classrooms of artists and designers with biologists, and we ask them to imagine, create the future of biotechnology. You're looking at some of the projects here. At the end of the semester, the top teams from each of the institutions come to the Museum of Modern Art in New York City, and they present their ideas on stage in front of judges that range from across biology, design, art, museum curation, entrepreneurship, social science, you name it. We've been doing it for about 8 years. when I think about what I've been doing for these last 15 years running these two nonprofits, um, uh, I have a lot of questions. Basically, the question I ask myself is, I'm not a scientist, I'm not a bioengineer, and yet for the last 15 years, I've devoted my life to, in some ways, evangelizing biotech. I'm going to air quote evangelizing, um, and and I've been thinking about why I've been doing that. And I'll give you the first reason. I have two today, and I'll give the second one later. But the first is that we non-scientists need to explore biology. We need to be familiar with how to read and how to write DNA, because we need the lens through which to look at the world. So basically, what I'm saying is that we need biology as a lens through which to look at our world. It doesn't need to be the only lens, but it needs to be one of the lenses that we have. So I grew up in a religious household, and I was surrounded by people who prayed all day, who spoke to God on a daily basis. But I would say that my first real religious experience was not in the classroom. It was when I was 30 years old, and I was with a group of friends. I had invited them into my house. This is the first meeting of our community biology lab before there was an actual lab. And we were basically hacking webcams to turn them into microscopes. And we took a little droplet of water and put it under our little microscope, and we saw this entire wild world, sort of wonderland of microbiota, little creatures zipping around. I remember this very clearly. They were like jelly bean-like creatures flying through the screen. There was this kind of toothy creature that was kind of chomping at the other creatures. There were paramecium with their little tassel-like flagella sort of moving about, and it was sort of this gorgeous moment, right? I had never seen anything like this. And it was literally in a single drop of water, literally like on the head of a pin. The irony of that moment is that I looked up to talk to one of my friends, and the light under the microscope is quite hot. The droplet had evaporated in that very moment, and that entire micro universe had just vanished. And so, in some ways, I had looked at a world, and then, of course, I destroyed that world at the same time. The irony, of course, is that when you start to see that, you realize that, hey, even as I look up at the lie in bed, there are organisms living on my eyelids, there are organisms living under my head and my pillow. They're everywhere. And so one of the reasons why I think that we should be thinking about biology is because when you have those tools at your fingertips, when you have the tools of the molecular biology of DNA, when you have the tools of microscopes, you can start to see the ways in which we're connected at different levels across the spectrum of organisms. So from the micro to the macro. And you start to see how all of these are interconnected. You can't have one without the other. They don't function. And I'll give you an example. So taking my notes out because I lost my track. So when you remove one of the rungs of life, so you're looking at a mouse that is called a no biotic mouse. So it's basically a mouse that has been born in a sterile condition, has never been exposed to any microbes. So there's nothing living on or in it. And this is an extremely sick mouse. So its intestines aren't working. The microbes in the gut that usually produce vitamin K and B12 are not there. The mouse has a messed up lymphatic system. Its intestines are swollen. When people talk about the dangers of germs, there's the reverse, which is the dangers of not having the germs living in and on our bodies. So we need to think of ourselves in a bit of a different way. We need to think of ourselves as an ecosystem, as part of a whole. So with the right tools, everywhere you look on the planet, life is thriving. So this is a thermal vent at the bottom of the ocean. It's never seen a plant or the light of day, and yet it's thriving with life. And in fact, some people believe that this is the origin, places like this is the origin of life on this planet. And if you look at a place like this, which is a scalding hot acid pool in Yellowstone, it's also thriving with life. And actually, the bacteria that scientists pull from this through research discovered a way to basically multiply DNA. So almost like make a DNA copy machine. And it's that exact technology that has had huge implications across the way we do things. So if you think about CSI or like these kind of cop dramas where they take DNA evidence, or you think about all these people who have been imprisoned and have been exonerated using DNA evidence. Without us looking at the organisms in pools like this, we wouldn't have had the technology to be able to actually do those things. So if you want to begin to appreciate life and the world in all its fullness and complexity, you both have to look at the micro level, appreciate how it weaves together with the macro level. And you can't have an individual life without the living environment and habits, and you can't have the living environment without all the individuals working in patterns that we're only now beginning to discern and understand. So you artists, designers in the room, I implore you to look at these patterns and appreciate them. I'm going to show you another image. So this, I would say, is my second most spiritual experience in my life. You're looking at a petri dish of bacteria that, again, me and my friends in our home lab grew. We genetically engineered that bacteria. It is a bacteria that is a descendant of a E. coli that a scientist roughly around 150 years ago pulled from literally the bottom of a baby. And we've been using that as a model organism since. And the reason why we do that is because it just grows like gangbusters. So if you make a tiny change now, within 20 minutes, it'll double and then double and double and double. And, within 24 hours, you'll be able to see the results of your experiment. Our experiment was basically to take a gene from jellyfish and put it into that organism, put it into that bacteria. And voila, we got a green fluorescing bacteria. So why is that spiritual? All right, it's because so the same DNA that codes for that bacteria to do the things that bacteria do in a baby's intestine, to have their little flagella, which you see sticking out there, is the same DNA that makes a strawberry red and have seeds on its outside, is the same DNA that makes me late at night crave Ben & Jerry's and want that fat and sugar. We're all made of that same stuff. We all have that same DNA, and it's the sequence of the order of these base pairs within that DNA. Those are what differentiate us from these other organisms. So that's both exciting and terrifying at the same time. So here's a little experiment I did. Between us humans, so you're looking, this is a side of Tolstoy's War and Peace, which is my favorite book. But if you were to take each letter in our DNA and put it in that book, that thin line that's about 7 pages out of a 1215 page book, that 7 pages would be the difference between you and me. So we're almost exactly the same. We are deeply, deeply related. I love this. This is another fact that I really love. So this is a banana. So if you took the DNA in a banana and you compared it to our DNA, about 40% of that DNA would be different. 60% of us are the same. So 60% of us are bananas. And that's, well, I'm bananas all the time. But that's sort of amazing and also like deeply troubling. And I'll show you why. Because if you eat the banana, you're also eating your relative in some strange way, which is which is cool and exciting. And so going back to this, like with the tools of biology, this is by the way my like attempt at some of the art that you can pull from, you know, typing words into the internet. But basically what I'm trying to say is that, you know, we're all intertwined on this very deep level, and understanding that and finding our connections to these other organisms, our connections to these living things on this planet is, I think, for me personally, what makes me feel connected. At the same time, it's also something that allows you to insert a gene into any organism, in this case bacteria, and get a bacteria to be photoreceptive and act like film. You can also use that genetic engineering to change a bacteria to produce human insulin to treat diabetics. Similarly, you can have a vat of yeast that produce beef proteins and have your veggie burgers taste like meat. And finally, you can take genes from one organism, mix it with a second organism, and take CO2 from the environment and turn it into things like clothes. At the same time, you can take a gene, tweak a human being or tweak an embryo, and have someone who feels painless. Or you can tweak a gene in a human being and have them need only 4 hours of sleep. Or you can tweak a human being and make them immune to HIV. Or you can literally eradicate a species. All of these are using the same technology that connects us to all of the other organisms in our world. That's a lot. So this brings me to my big number 2, which is not only do we need the tools of biotechnology to deepen our existence and deepen our understanding of the world, we also need, biotech also needs us to make sure that we're using it correctly. I see that I'm running out of time. So I'll be very quick to say, you know, we are obviously encountering a major environmental crisis. There are dozens and dozens of potential applications for biotechnology. We are facing a crisis. We can use it for good. We can use it for bad. It is a big ethical question that we all need to decide together. And it's not just the realm of scientists or business people to make these big decisions for all of us. It's really the realm for all of us to make these decisions. So again, I implore you, biotech needs you to help make ethical decisions about how we're going to use it in the world. So very quickly, as I pass the baton to Elizabeth, we need biotech. We artists, designers, regular people need biotech because it deepens our understanding of our world around us. And number 2, biotech needs us to, 1, reach its potential, 2, avoid the terrible pitfalls that are involved in harnessing any kind of technology, and 3, finding the ethical applications to go forward and hopefully make a better future for all of us. So I'm going to now hand the baton to Elizabeth. Thank you, everyone.
Thanks, Dan. I feel like I'm very familiar with your work, but it's always awesome to hear you put it together in a talk and kind of see how you stitch together all of those threads. I'm really happy to be here with y'all to share some projects that are very much related to what Dan just introduced. And then what we're going to do after is we're going to do a fast round of critique of some of our favorite art and design projects to share with you and tell you what we think about them. So I'll go ahead and share my screen. Can you guys see white text on a black background? Awesome. So as Carlos mentioned, you know, I'm trained as a scientist, as a computational scientist, computer scientist, and also biologist. And Dan says that, you know, he founded some nonprofits, but actually I'd like to amend that definition and say that you founded 2 communities. And I largely credit those 2 communities for kind of queering my perception of biology to understand that biology is actually a lot bigger than how we define it in like the canonical academic institutional biology lab. So I moved to New York City in 2014, quickly got hooked up with Genspace, and my mind was just blown. I was like, wow, there's people who are doing like really rigorous, straight up molecular biology in a lab that does not belong to any institution, that doesn't have to answer to any institution. I mean, you guys had to answer to the FBI, right? But not like an academic institution. And so I really credit Interaction with, Genspace and its members for really shifting my perspective about how I do biology as a scientist and becoming really interested in how biology intersects with society. So how is it meaningful? How are the things that we do in the lab become meaningful for outside of the lab? And then time went on, and Dan found this other amazing community, the Biodesign Challenge, which I got super stoked about and have been taking student groups there for a number of years now. And that was kind of the second querying experience that I had, where I was like, oh my gosh, art and design are equally, or can be equally, rigorous methodologies as the scientific method to approach questions that are meaningful to the sciences, that are meaningful to the biological sciences. And so what I'd like to do now is just talk, about a couple of projects that I run in my lab, but specifically kind of how I envision the research framework in my lab that I run at NYU. So my lab is interested in environmental microbiomes, so populations of microorganisms in the environment. And the reason why we're interested in environmental microbiomes is because, as Dan mentioned, there's this growing body of knowledge that describes that multicellular organisms, so like plants, animals, human beings, owe many meaningful aspects of their growth and development to the microorganisms that they interact with, that we live in symbiosis with. And so just taking the example of the human microbiome also interacts with and is impacted by the environmental microbiome. So the microorganisms that inhabit the environments that we live in and work in and transit through. And for those of us who live in cities, which is more than half of the global population at this point, the environmental microbiome is sculpted by design decisions made by design professionals, architects, material scientists, city planners. So if you were able to figure out all of those pairwise interactions, so how does human health relate to the human microbiome, and how does the human microbiome interact with the environmental microbiome, and how do design decisions impact the environmental microbiome, you could string together all of those pairwise relationships and design probiotic buildings or probiotic cities. That is a great big blue sky endeavor that is, not the job of any one single researcher or one single research lab. And what my lab focuses in particular is that last pairwise interaction. So can we identify relationships between abiotic aspects of the environment, and so non-living aspects of the environment, and its biological component or its microbiological component? So at my lab, we kind of have a portfolio of projects where we find kind of natural experiments within the city, kind of use case studies within the city to start to establish relationships between kind of aspects of the built environment and its living component. And we don't only use scientific methods for that. So we really try to use a multimodal methodology to try to answer those questions, both from a scientific perspective, from an art and design perspective, and also from a scholarly perspective. I get to work with a really rad group of students in my lab from a whole range of different backgrounds. So we've got Phoebe and Deidre, who both have backgrounds in architecture, and Deidre's in the civil engineering department. Harry, who's a molecular scientist, Sheila and Anshika, who are biomedical engineers, Howey, Proga, and Andy, who are biomolecular engineers. Andy also has a double major in fine art, which is awesome. Tori is a major in public health. We have Charlie, who's doing bioinformatics, and Caroline, who's coming from the humanities department. So we've got a really wide range of students in the lab, and I feel really fortunate that I get to think with this really rad group of people. And so the way that we envision this is that interdisciplinary work means a diversity of outcomes. So you can take this one thing that you're interested in and approach it from many different directions. You can approach it from the scientific perspective, so designing controlled experiments to be able to understand it. You can approach it from the creative perspective, so making critical art pieces, for example, to try to understand how this thing that you're interested in relates to society. You can also approach it from a scholarly perspective, so trying to use tools of the humanities, feminist theory, philosophy, to try to understand how this thing that you're interested in is interacting with, you know, previous scholarship. And so I just wanted to go through quickly just a couple of projects that are just, I find to be our hallmark projects about how we can use these interdisciplinary methods. So this is a project sparked by conversation or started in Genspace by 2 Genspace lab members. And it has now taken, you know, we've been working on for more than a decade, but here we're looking at an anthropogenic contaminated environment in Brooklyn, New York. So this is a contaminated waterway, and we're looking at how microorganisms respond to the anthropogenic contamination. It turns out, not surprisingly, that the microorganisms that inhabit this contaminated environment have evolved molecular mechanisms to degrade the toxic compounds that they've been challenged with. So from a scientific perspective, we do a bunch of field work. We paddle out in hazmat suits to collect these samples of contaminated sediment at the bottom of the canal, publish articles on that. But also I wrote a book chapter in an edited volume that was put together by a landscape architect that was focused around material agency. So how do inanimate materials actually have agency in our societies? And so the kind of perspective that I was taking there is that any kind of inanimate material that we encounter in our world has likely been metabolized at some point by a microorganism. So microorganisms are transforming our materials all the time. And so as such, any kind of material that we can observe is a certain sense of a history. It's actually a history, physical history of a relationship. So I kind of wrote about that from a theoretical, like, material agency perspective. This is an ongoing project. I made a zine called the Gene Zine. This is the first of a whole series of zine publications. And so what's particular about this gene is that it has DNA from the Gowanus Canal embedded in it. And so if you think of zines as like serving a subculture, serving as a communication mechanism to a particular subculture, this is a genomic note from the underground serving the subculture of multi-species collaboration. We recently had an art show here in Brooklyn over the summer that featured some kind of macroscopic photography of microorganisms and algae that we grew in the lab, that were growing out of this contaminated sediment that from a human perspective is uninhabitable and a forsaken environment. But from a microbial perspective, if you can kind of translate that, do that translation of scale, is actually quite a thriving ecosystem. So that's just one aspect of how we kind of bring to bear these different methodologies. I think it's important to note, though, that here I put kind of bidirectional arrows, because I think that it's important that each one of these methodologies inform each other. And oftentimes the relationship between science and art and design is a unidirectional relationship. So the idea is that, like, science is the knowledge generation methodology, and then art and design is either the critical or the illustrative methodology. And oftentimes when you think about collaborations between artists and scientists often imagine that the role of the artist is to make the science look pretty or to kind of illustrate those scientific concepts. And so what I'm really striving for, which is actually pretty difficult, is to have that go in both directions. So how can art and design practice actually help with, for example, hypothesis generation in the sciences to help us generate new hypotheses that then we can test with the scientific methods? Another kind of hallmark project that I'm excited to share is where we designed like a collaborative sampling methodology to be able to sample microorganisms from the air, but collaborating with honeybees. So we developed this whole methodology where you can collect debris at the bottom of a beehive and use DNA sequencing analysis as a way of understanding what are the microbial contexts that the bees have been traversing on their foraging journey. So we published a paper for that last spring. Andy, Sheila, and Tori are the lead scientists on this particular project. They developed this novel DNA or they prototyped a bunch of different DNA extraction methods. So when we're using DNA sequencing as a way of knowing, we need to make sure that we don't introduce, or at least we know the bias that we're introducing in that process. So they did a really exhaustive prototyping process to understand what the best DNA extraction method was. So here's Tori in the field, Sheila in the lab. They also presented a design object and an art installation at an exhibit in New York called Data Through Design in 2023. And and also presented at, oh yeah, we had a lot of fun doing that exhibit. It was great. And they also presented at the Biodesign Challenge in 2023. And I'm really proud to say that they were awarded the Science Sandbox Prize for public engagement for the amount of work and thought that they put into public understanding of science and how that project can kind of translate scientific concepts to the general public. And with that, just to conclude, I'd like to kind of bring home this point that to me, one of the critical roles of design in the practice of biology is that design focuses on defining interfaces and interactions. So designers have thought a lot about how does an object or an act mediate a particular interaction. And this is important because here, this image that we see on the left is a swab that we're all uncomfortably familiar with after several years of having swabs stuck up our noses during our global pandemic. And so this particular object is designed to collect a microbial sample. And it is designed in a clinical context. And we have very specific kind of affective relationships to it. So if you see this swab, you think contamination, you think pathogenicity. It gives a particular kind of negative affective relationship. There was one time that we were doing field work in the subway, collecting swabs from the subway. And so people, one of the teams was challenged by someone who thought that the team was implanting HIV in the subway. So they were reacting to this clinical tool and perceiving it from a bioterrorism standpoint. This was before the pandemic, so before even viruses were on people's minds. But anyways, this tool is designed to collect a biological sample. And then the image on the right is doing exactly the same thing. So grabbing a handful of soil is also collecting a biological sample. In that handful of soil, there's going to be billions of microorganisms that you can collect in that manner. But we have a completely different relationship to the action on the right. So the action on the right can be described using positive terms like groundedness and connection, and there's no kind of fear associated to that image on the right. And so what I think is really important is that oftentimes in the biological sciences, we don't have direct relationships with our organisms of study. So in particular with microorganisms that we cannot see with the naked eye, our relationships are always going to be mediated by an instrument. And so in that sense, our relationship really is to the instrument, less so than to the organism of study. And so, we think of scientific instrumentation as kind of this unbiased extension of our sensory system that allows us to perceive things that are otherwise beyond our senses. But that's actually not the case, right? So the author Karen Barad has written a bunch about this, if you guys are interested in reading more. But our scientific instruments are very much imbued with our biases, our hopes and dreams, our expectations for the outcomes of the experiment. And so for me, I see that the role of design is thinking about how meaningful those interfaces are that we create to allow us to interact with biological organisms. And so with that, I think, to me, the practice whatever expertise you're coming from doesn't particularly matter. It does not necessarily impact the types of questions that you can work on. So we tend to think of maybe like science asks these kinds of questions and art and design asks these kinds of questions. What I really feel is that we can all ask the same types of questions, but we're bringing different methodologies towards those questions. And those methodologies also come with different types of outcomes. But ultimately, it's all research around the same question. Outcomes, might be books, they might be creative works, they might be journal articles. And there's been kind of, I think, an unmet need for hosting those types of outcomes that are at the intersection of methodologies of science and design. That's an unmet need that the Biodesign Challenge has started to answer, for example. But I think that there's also an unmet need with respect to publication outlets. So peer-reviewed publications that will publish projects that are at the intersection, that use intersections of these methodologies. And so I'm going to do a shameless plug because I am on the editorial board for a new journal out of Cambridge University Press called Biotechnology Design. And so the goal of this particular journal is to publish projects that are at the intersection between, or that use methodologies that are at the intersection between the arts, design, and scientific methodologies. And so the way that we've framed this publication is around a set of like research questions. And the one that I'm hosting, and I'm really glad to answer any questions about, is called Vernacular Biotechnologies. So kind of putting forth the idea or challenging the idea that biotechnology belongs specifically to modern Western science, and asserting that biotechnologies have been implemented through traditional ecological knowledge, and also by people who don't necessarily identify as scientists. So biotechnology happens in the field, it happens in agriculture, it happens in the kitchen, and I'm glad to answer more questions about that. I teach classes on all of these topics at NYU, and obviously all of these projects happen collaboratively, and I'm very grateful to all of my collaborators. And next, we have our new TV show that Dan and I are going to start to host, Hot Picks with Dan Grushkin and Elizabeth Hénaff.
There's no TV show. That's a joke. So Elizabeth and I came together and wanted to pick 3 of our favorite projects that we've seen over the last couple of years or over the last decade. And we're going to show them now. So let's go to the next slide. So here's a project that I love, and I actually think it's one of the sort of masterpieces of the world of bioart. It's a project called Resurrecting the Sublime by Daisy Ginsburg, who's an artist who's been in the sphere of synthetic biology for a long time. She worked with a scientist at a company called Ginkgo Bioworks, where they went into the Harvard Library's how do I say it? Basically, collection of lost species of plants. They identified plants that have gone extinct in the last 100 years. They took DNA samples from these plants. So I think this is a plant from Hawaii that produces quite a fragrant flower. But of course, no one has smelled that smell in over 100 years. So by basically analyzing the DNA, they identified some key proteins that would produce scent. And in some sense, this is a project about what you can't do with biology. So although you can produce all the molecules that a plant might produce in growing, you don't know what proportions, you can't imagine what you can't know what it would smell like in that environment. And so what they did was they took these smell proteins, put them into a concoction. You see it in that box of a room over there. And they have different combinations of those proteins being sprayed into the room. It's kind of a perfume exhibition. And then, of course, there are environmental parts of that and pieces of that environment in that room as well, to try to give a context for what it might have been like to be in that field with those flowers But of course, we can never know. So you get all these variants of smells depending on what time of day you go into the room and what combination of chemicals or odorants they put into the air. I don't know, there's poetry here that I'm not quite expressing, but I just think it's a masterful way of using DNA sciences to learn something about a world that we can't actually recreate.
Yeah, I think this project is also particularly beautiful because it makes us think about our other senses that we use to perceive the world, and we're very visually dominant beings. And so when we think of environments, when we think of interacting with environments, we really think about our like visual system. But this one is really challenging because it's asking us to use a sense that we rarely use kind of intentionally and consciously. And I've seen various different installation formats for this exhibit. And this one portrayed here, there's a screen that shows visual material, but in other formats of the exhibit, there's actually no visual material. So there's, the same kind of container box and then the stones that you sit in. But it takes you a moment when you enter the exhibit to actually understand what it is that you're perceiving. And I think that's a really interesting treatment of that like relationship to other organisms, non-human organisms. This next project is called Eggstro Eggs. And it's a project by Mary Magic. So Mary Magic is an artist who has been working in the bioart field for quite a long time. And they've done a bunch of like really seminal artworks in the bioart realm, but this one I think is particularly interesting because it really challenges the idea of, or it really critiques the idea of our relationship of domestication to more than human or non-human animals. We've domesticated so many species for our use. And the premise of this particular project is that these chickens have been genetically engineered to produce hormones that are useful to human populations. And so this particular project is describing it as producing hormones necessary for fertility hormones. And so I think it's particularly interesting because it feels really uncanny to domesticate a species for their production of this molecule that otherwise seems that it should be kind of a proclivity to a human body. And, our hormones in particular, our sex hormones feel very like identity defining. A lot of the work that Mary does is around like trans rights and how rights to hormones that express your own gender identity should be like a human right. This is particularly difficult in the United States. But I think that this project, as far as critical projects go, has just so much to say about our relationship in society with respect to more than human organisms, with respect to the process of domestication, and also with the process of like hormones as defining identity. I don't know, Dan, if you had more stuff to say about that one.
Yeah, I guess, I mean, I think that there's kind of in a lot of bioart, there's an appreciation for the grotesque. There's this sense that, we're mixing our own genetics, our own hormones, our own enzymes, chemicals with other organisms, and they're producing them for us. Like you see this a lot when people talk about xenotransplantation, where they are genetically engineering creatures like pigs to produce human organs, which is sort of the cutting edge of research these days. And there's kind of a grotesque factor to it. And yet at the same time, it could be life-saving. At the same time, it could be incredibly empowering. But it also speaks to the strange multi-layered relationship that we have with the organisms that we domesticate. And and on one level, they're a technology, they're a tool that we use. On another, they're an organism with their own rights, their own right to exist and to be, and we have to grapple with that as we work in the sciences and work with them. So that's, I think, something that we as humans, since I think the domestication of animals and have to contend with. And I think as we understand them more deeply, with the tools at our disposal, the more we'll have to grapple with those questions.
Yeah, that and the power dynamics involved too, in domestication.
Sure. This is my hot pick number 2. I know we're running short on time. So this was a project from Biodesign Challenge 2019 from a group in UTech Sydney. They were looking at some of the cutting edge in what you can do with DNA. And there was a lab study, or there was a study that came out of Harvard University where the scientists were able to show that they could store data in DNA. So by changing up the sequence and sort of putting it to a specific code, you could literally embed videos in the DNA of an organism, which is a really crazy thought. The students started to think about what are potential applications for that. And what they came up with was surprising. They started to think about what the future of the cemetery might look like. And they imagined that instead of having gravestones where you have some markers about the person who passed away, you could take elements of that person's history, their memories, photos, videos, songs they liked, whatever you thought was, something that would memorialize them, code that in DNA, and then literally insert that code into a plant or a tree in the environment. So imagine entering into a grove and all the trees are sort of memorialize people who have passed away. You take your little DNA sequencer, pull a leaf, and then be able to read all these memories about the people who have passed. A really strange, surprising way of using this new technology. And also poetic.
Yeah, and if you think about it, genomes are already kind of records of what has happened evolutionarily speaking to that plant or to that organism in general. We talk about genomes as being palimpsests, a substrate that has been written over and over and over again, and those layers kind of are a record of the history of evolution. What's interesting to me about this project is also leaning into the idea that DNA changes over time. And so this memorialization is also something that's still alive in a certain sense, because it's still changing over time with the just kind of background mutation rate that happens in DNA in general. This next project was a pick of mine. So this is a project that was presented at the Biodesign Challenge in 2018 called Minima. And what I really liked about this is that it's an illustration of what I was just talking about before, about how the design of the interface or the tool matters. So this project was designing a isothermal PCR reaction that allows you to detect the presence of genetically modified organisms in your food. So this is, a high-tech biological reaction that if you were to do it in the lab, it would involve like those white plastic test tubes and pipettes and kind of all the like recognizably laboratory apparatus of the laboratory. But the way that these students designed this is that every aspect or every tool necessary to perform this reaction felt like kitchenware. And they were designing this because they were like, okay, if you're going to be doing this experiment in the kitchen, it might feel kind of incongruous or uncanny to be using stuff that looks like labware, like plastic tubes and stuff in the kitchen. So let's design everything to look like kitchenware. And so they really thought through the process and, made this cool video where they explained like everything, is dishwasher safe. And so you can just put it in the dishwasher once you're done doing your testing, just along with your, teacups and plates. And so I think this project is a really beautiful illustration about how design can really like shape the relationship to a biotechnology by thinking carefully about the kind of product design of the objects that actually implement that biotechnology.
I'll add to that. There's something, elegant and beautiful about this as an object. But it kind of reminds me of those pictures that I showed you of our early days in my kitchen doing lab experiments. And there were 2 experiments really happening at the time when we did that work. It was the actual, are we actually going to make a genetically engineered machine, organism? Are we going to be able to, see DNA or amplify DNA? But the other experiment was like, can we do this in spaces that aren't the laboratory? And what I love about this project is that it embraces really sophisticated science that took us, this particular science won the Nobel Prize, but took a lot of human ingenuity and hundreds of years of, or 100+ years of research, and they've made it into something that you can do at home, and it's something that feels like cooking, right? Like, I love that they're using a tea light to do that, to warming their reagents. Like, I think it's a lovely project, and I appreciate you bringing this one up, Elizabeth, because it's been a while since I've seen this one. All right, this is gonna be hard for me because this is Elizabeth's project, and I have to describe it. I'm gonna flub. I love this project, and I cite this project all the time. I should also say that, Elizabeth's work, her participation in Genspace from the earliest days to her participation in Biodesign Challenge has deeply, deeply influenced those communities, and has deeply influenced the way that we think about literally the living world around us. So I just have to thank you for joining these communities when we frankly had no idea what we were doing at the very beginning. So, all right, here we go. I think I softened you up. So, so here's a project: subculture microbial metrics in the multi-species city. This is a project that Elizabeth did with Kevin Slavin and The Living, which is an architecture group, and they created a surface at the storefront for art and architecture that would inculcate the living microbes in the environment. So what I love about this project is that they weren't just designing for humans. When you think about architecture, it's about habitation, it's about people, it's about where they're going to go, where do you put the various facilities to them. But Elizabeth and co were thinking about, what about the other organisms in the environment, in the neighborhood? How do we design for them as well? And so this surface is sort of attractive to the local microbiota that would settle in these crevices and start to grow and thrive, hopefully. And then what was wonderful about it is, because they're invisible to the eye, because they're invisible to us, without our tools of biotechnology, you can then, or the team could then go and sort of take their swabs and take little bits of bacteria and other organisms that have settled in this environment and come to know their neighbors in a totally different way. I love this project. But yeah, Elizabeth, I butchered it. Go ahead.
No, not at all. This was such a fun project to work on, with really awesome collaborators. And yeah, I think that, thinking about multi-directions between science and design, the prompt that was given to us by Eva Franken-Girabert, who was the curator at Storefront at the time, was, what would it look like on an architectural scale if you designed for microorganisms? So that was the prompt for the commission. And that's how we ended up, thinking about, okay, we need to design something on a human scale that exists and has a function on the human scale. So designing a facade for this gallery space, but also it needs to have an interaction with the local microbiome on a microscopic scale. And so that informed like the material choices, which was this kind of sandblasted wood as a bioreceptive material. So what I really like about this project is, for me, it was one of those times where the arrow went in the other way. So there was like a design prompt or challenge that actually made us think about the biology in a completely different way.
And I would say that this has become one of the principles, one of the fundamental principles around biodesign, which is that you're not just designing for us humans, you're designing for the entire living spectrum all around us. And that is a huge responsibility and a huge task. But the more you understand about our environment and the organisms around us, the more thinking you have to do about what you put into that environment. Yeah, kudos to you guys. That was a fantastic project.
I mean, one day we'll have a Rhino plugin where you can like parameterize the microbiome that's going to inhabit the space that you're designing. This next and final project that we wanted to share, it was one of my picks. So this is a project by the artist Pierre Huyghe that was presented in Munster in 2017. It's an iteration on an idea that he had presented earlier at the Paris Tokyo, but this was a project that I had the chance to work on as a science advisor, like bioadvisor, for the Huyghe team. And so Pierre Huyghe has had a long career of making artworks that involve more than human organisms. You've maybe seen the works that he did with bees, or also he had an exhibit at the Serpentine where he released like a bunch of flies into the gallery and let the flies do their thing. And what this project and then the project at Paris Tokyo that was called Living Cancer Variator had in common is that there were human cancer cells that he had installed in an incubator that and their growth patterns were influencing other parameters of the exhibit. And the reason why I like Pierre's work so much is that I feel that he gives agency to the more than human organisms that he incorporates into his exhibits. And I think that that's actually pretty outstanding because a lot of the practice in the sciences, but also I think in the bioart field is having a preconceived idea of what your organism is going to do in your project. So whether it's in the lab and we have vocabulary around that. We isolate things, we purify things, we don't want things to be contaminated. There's very much kind of like a power hierarchy of control implemented in this. And I think that's also the case for the bioart that we see in galleries. You put an organism somewhere and you want for it to do XYZ, and you kind of build infrastructure around that to ensure that that happens. And Pierre works in the completely opposite way. And so he kind of sets up these ecosystems and then he lets them do what they want. So if the flies are going to die, that's fine. And if the bees are going to leave the hive that was created for them in the sculpture, that's also fine. And I think that this is a really important concept to kind of hold on to as in your practice of art and design, or as artists and designers, if you're thinking about working with more than human organisms, is how can we give agency to more than human organisms who maybe don't have the same ways of communicating as we do, but give them agency in these works to allow them to actually be expressive and kind of flip those power dynamics that otherwise we've implemented over a long period of time. We've mentioned domestication and everything like that. So that's why I particularly like Pierre's work.
I think it also calls attention to this really interesting problem that you have in exhibition spaces, right? Usually galleries and museums have precious pieces of art. They're doing everything in their power to keep those pieces away from living things, to keep those pieces away from the insects that might eat them, or the fungus that might chew on them, and so on and so on and so on. So when you introduce pieces like this, or like the Fly project, you're introducing a kind of a chaos into the gallery space that a lot of people work against. And I think that's really interesting in challenging what we can have in these spaces. Yeah, I appreciate these works as well. This is a new one to me, by the way, Elizabeth. So thank you for sharing it.
Yeah, sure thing.
I know that we're running a little long, but we're happy to take questions. Feel free to.
Yeah. Let me see if I see any questions within the public. If not, we can continue and follow up some topics. And I don't know if there is a question. Let's see. I think that maybe
Ram has a raised hand. And
Hi, audible. Sorry. Okay. Thank you again, Elizabeth and Daniel. That was so much fun to see a lot of the projects and possibilities that we could go ahead with. And in fact, one of the questions that I did have was about this idea of like a lot of the times using these organisms, and which, by the way, I love. I think it's very endearing that you guys use the word more than human organisms. It was really nice. But the fact that like a lot of the times that they're predictable, and how do we sort of give that agency? But then you guys already answered that with the last slide. I was being a little snoopy with your open tabs, Elizabeth, and I happened to already like look into the vernacular biotechnology thing. So could you speak a little bit more about that? Because as I understand it, it's still an open project that you're inviting things into, maybe like an example of something.
Sure. So it's an open call for papers. And so we invite different types of papers. Those can be descriptions of a case study. They can be like think pieces or a call to action. It's there's different kind of modalities that we're open to publishing. So you can think it kind of like fits the gap between the like straight, like the novel research science publication and the humanities essay. So it's kind of like that intersection space. And so we organize them around different research questions. So if you visit the website, you'll see that there's a bunch of other different research questions. There's like, can we grow a building and why would we want to? Or living textiles. So there's a bunch of different research questions. And so vernacular biotechnologies is one research question. And so the call for papers there is inviting publications that describe processes, practices, rituals, positionalities with respect to, or I guess pushing against the fact that the term biotechnology is very much appropriated by Western science, or specifically modern Western science. And so, you know, there's this kind of generalized idea that biotechnology is as a field is a product of kind of the modern molecular methods that have been developed in maybe the last 50 years. But defining kind of interactions and relationships with organisms in the environment and for like human use is definitely not a new thing. And so we're inviting proposals from people who maybe don't identify as a biological scientist and who are doing biotechnology in non-canonical spaces. So like the kitchen or the field.
Awesome, thank you.
Sure. If any of you have questions about ideas that you wonder whether they would be fit for submission for that publication, I'm really glad to answer those questions offline.
Yeah, I just want to jump in and just say, you know, this is something that we've been working on for quite a while and trying to figure out how do you know, combine something like a design monograph with a scientific paper. I think that the work that Elizabeth is doing with the journal, I think I'm still on the advisory board, is really important, because you don't want just when you're designing for the world, or when you're thinking about how you're going to implement these technologies in the world, you don't want it just to read like a scientific paper. You also want it to be a design monograph. You want to understand why people are doing what they're doing in a deeper way. And so finding the balance between those two, I think, will be the next decade of work for that journal. And I'm super excited to see how it evolves.
I think that we have a question. I think that Vivek is.
I don't think I can't hear you.
Sorry. Because like a speaker is open next. So I got confused. Sorry. So my question, I have a bunch of questions, but I'll try my best to probably view them so that they kind of are more take less time, hopefully. Where should I start? Okay. So the first thing, when you were talking about when you were using swaps, in the metro, and people thought that they associated to bioterrorism, And this is one thing which I really love this quote by Wilson, and I like to throw it around everywhere. It's like we have Paleolithic emotions, we have industrial institutions, and we have godlike technology. And specifically when we live in an information war, sort of when cultures are getting really divided, and like we have extremes, and like people are getting polarized, how do you imagine a science which like which is as egalitarian and as appealing to everyone, and doesn't like like just, I mean, I don't even know how to put it in words, but like which can bring everyone together, because everyone is trying to either reject certain scientific ideas, or they have a complete rejection of the system itself, or someone is like really holding on to their personal belief as compared to what we can achieve from evidence-based science, as I have learned as a kid while growing up. So because you are directly you're working on the ground as science educators, and I'm sure you come across this as a challenge again and again. How do you navigate in that world, if that makes sense?
I can give you my personal thought about that, and I'm sure Dan has ideas about that too. I think that polarization maybe happens actually when we try to define a grand unifying framework that fits for everybody. And to me, I feel like an important thing about science is acknowledging its limitations as far as what kind of knowledge it can generate. And that also can be many different ways of generating knowledge that they do so in different arenas, right? So science is really good at generating knowledge about things that are measurable and things that are repeatable. So can I measure this thing that happens either with my own senses or instruments that I've made? And can I repeat those measurements? Can I design experiments that allow me to repeat that phenomenon? And so that's those are the conditions for generating knowledge within the scientific framework. However, there's all sorts of knowledge, and that fits pretty well for all sorts of ways of interacting with the world. But there's also knowledge that is not measurable or is not repeatable. And I think that knowledge generation in the context of religion or spirituality falls in that category of non-measurable and non-repeatable. So like if someone feels like they had a religious experience, they won't necessarily be able to go back to that same place and have that same religious experience. And they won't necessarily be able to share some metrics of that experience with somebody else to validate it. But that doesn't mean that experience was any less valid than experiences generated through scientific knowledge. And I think actually, I mean, I'll speak here from the perspective of being a scientist here in the United States, is that I think that a lot of the polarization that happens is through trying to make one vision of the world or the other vision of the world fit the whole world. But actually, if we were able to accept the fact that science and religion are different ways of generating knowledge and perceiving the world, and that those two can be true at the same time, then I think actually we would experience a lot less of the conflicts than what we're experiencing at the moment.
Yeah, just to add to that, I mean, I think of science and these tools as a lens, right? But you have a lot of different lenses, and you can apply them at different times and look at the same thing with those different lenses and come to very different conclusions. And my argument is we should just have that lens at our disposal. It's funny because, having heard your question, I heard a different question as well, which is, what do we do about people who might use these new technologies in malicious ways? But did I mishear you?
I mean, they do have both the tails. Like, I mean, as I was saying, I want to condense like a bunch of thoughts in one question, but like, yeah, when I'm trying to weave it, I like both of these things concern me. Like, one is like maybe like to add another example for like uh, like politically how, for example, I come from India, and like science has become kind of a battlefield of reinforcing your cultural ideas. So you have this battle going on that, oh, this is Western medicine and we reject it. We will go for our home medicine or Ayurveda. But to a reasonable mind, it should be that science has no borders. It doesn't belong to West or it doesn't belong to East. So like my concern are on the both ends, like how, like someone using the godlike technology for all the malicious kind of ways, but also the differences being used to reject the good godlike technology which we might have at our disposal. So yeah, it's the both ends which puzzle me equally.
Yeah, I mean, I feel like we've been thinking about the biosafety, biosecurity issue since the very beginnings of our organization. You know, when we said, hey, we want regular people, non-scientists, to have access to the tools of science, there was a lot of pushback from both the policy world and also the media. And the stories they generated were Frankenstein stories. There was, you know, the story that, you know, if we give access to this technology, people are going to make drugs in their basement, or, you know, using like molecular biology, or they're going to make a super virus that's going to kill everyone, or, oh my gosh, the list goes on and on. And, you know, I spent a lot of time in the policy world knocking on doors, talking to security folks, saying, hey, look, there's a negative side to not, you know, and exposing people to this information as well. And saying, look, like, we're a learning community. Are you trying to, you know, shut down the sort of information that should be freely available? Isn't this information important for us to know? Shouldn't the public be informed about what actually happens in the lab and how it works? All of these questions. And so you're absolutely right. There are really scary sort of stories we can concoct about what one might do were everyone to have access to the tools of biotechnology. At the same time, you know, there are guardrails that can be put in place where access to, for example, I don't know, anthrax or some sort of really nasty, dangerous organism isn't publicly accessible, isn't widely available to everyone, right? Like, although I want everyone to have access to the tools of biotechnology and I want everyone to sort of appreciate them, that doesn't mean I want everyone to have access to, you know, smallpox in a vial, you know? And so we have to think really hard about what are those guardrails, and we have to agree collectively on how to implement them. And so, you know, the Geneva Convention has a law against bioweapons. Super important. You know, we've agreed as a nation, a world of nations, that that's not something that we're going to pursue. Those kinds of things are very, very important.
Thank you so much.
I don't know if there is other questions, but maybe in the meantime, I wanted to ask you, because both, no, that we've been talking about the question of the security, but there's also another question that maybe it's also very prudent today, which is connected to the ethics, no, of what are the limitations that the experimentations in laboratory, and what is the stand, and also how do you feel, you that are in the forefront of that sort of research, which also means pushing forward certain ways the limitation of certain ethics, no, within the realms of what can be experimented, and the means and the tools, and also the objectives, and what are these thresholds now, no, on the ethics about using and manipulating organisms, no, and different scales, no?
I can answer that from my own perspective. I think that there's there's different ways that you can think of it, so we have, so kind of from an umbrella perspective, there's federal law that dictates ethical guardrails around scientific research. So specifically that were put into the internal review board that was specifically put into place after really atrocious experiments on humans, vulnerable humans, were brought to light. So vulnerable humans, including people with disabilities, people of color, homeless people, all in the United States, as late as the 50s. So there's legislative guardrails about what kind of experiments are possible, and there's kind of federal institutional review that that anytime you're doing any kind of experiment on humans, you have to submit that paperwork to be able to have your experiment approved. There's also ethics. There's also legislation with respect to animal rights and how you can do experiments on animals. So as far as, so there's kind of like that, like legislative overview. But then I think also on a personal basis, you have to ask yourself what are the goals that you think are meaningful from an ethical or even moral perspective, and what are the goals that you want to like contribute your time and effort and like brain cycles to. And I think there's kind of, in the sciences, there's generally like a divide between what we call basic science and applied science, with basic science being the idea of let's just study things to understand how they work without necessarily a particular application or outcome of that research in mind. And then there's applied science, which is let's study things with a particular idea in mind about what it could be applied to from an engineering or technology perspective. And I think that there's value in both of those, and that the ethical questions are different in those two. So maybe like if you consider, say, an experiment on animals, and here I'm talking from my own personal standpoint, and I've had arguments with people on this standpoint, but I think that if you're doing, say, an experiment on animals that is that does not necessarily have clear benefits either to that, hopefully to that particular animal, or to maybe human beings, with like a well-defined population of the human beings who would benefit from that, then I don't believe that it's ethical to do those particular experiments on animals. There's a lot of experiments done on like monkeys, other sentient animals, to just kind of understand how the brain works or understand how vision works. And I personally, I don't contribute to that research. I've, decided to change career from human research that involved a lot of animal models to environmental research that does not involve animal models. So I have my own personal kind of moral compass with respect to that. Yeah, I don't know. That was just actually a ramble. It's a very long question. Yeah. Or very long.
I mean, I'll tack on to that. I think a lot of what we've been trying to do, certainly with Biodesign Challenge, is try to bring a group of people together from multiple different backgrounds, not just scientists, policymakers, ethicists, artists, designers, clearly, the communities that are going to be affected by these technologies in the long run, and have these conversations before these technologies get introduced. And really the whole purpose of the Biodesign Challenge was to bring those groups together and get them to talk and get them to see potential applications before their actual applications. And so, we're working with students who are presenting all these wild ideas about how biotechnology might be used. A lot of the debates that we have among the judges and the professionals involved are exactly that ethical conversation. And so I'd like to say that we're seeding it. I'd like to say that the fact that we can, choose our best projects or the winners of our prizes in a room of people with such diverse opinions suggests to me that we can also get people of widely different ideas about ethics and have them also make decisions about what technologies go forward and what don't. It's almost impossible to actually have this conversation without starting to bring up all the exceptions that we've seen in the last couple of years. So, you know, I kind of mentioned He Jin-Kyu's sort of research work where he introduced a genetic mutation to embryos, and two babies were born that you know, presumably may have some resistance to HIV. It's not clear. You know, that was widely derided by the entire world, by certainly the entire scientific community. He went to prison for a couple of years for doing that. There's a great New Yorker article about him that just came out in September that talks about where he is now. But you see that people, you create ethical rules or accepted guidelines, and then people will break them, and that's something that we have to contend with as a society.
Yeah, I think it's important work that's being done, you know, like you mentioned, in the, you know, in the judging groups. But I think that also at the same time, it shouldn't be the role of others to be watchdogs of the practice. And I think that that's a really tricky question. You know, how can you kind of maybe bake into scientific education that ethical component? Because in science education, you know, you learn like the what and the how, like what can I do? What are the technologies? How can I, you know, how can I execute these particular experiments? How can I achieve specific goals? But the like, why is it being done and who is being impacted is kind of tacked on as an afterthought in most educational programs.
Which is wild to me, because I think that's just like, that's the work of being a human. Like, why do we do the things that we do? Like, this is the sort of fundamental question of our everyday life. The fact that it gets tacked on in scientific research is a real problem. And I would also,
it all boils down to capitalism, right? Like, what is paying the money and what are people able to allocate time to?
Yeah, I mean, if we add capitalism, which I guess which has been like sort of the elephant in the room when you talk about technology, yeah, you're contending with a lot of forces, and you know, I want the moral forces to be the ones that win.
Great. I think that maybe we have time for a last question. I think that Dora is.
Hey, Dora.
Hi. Thank you. It's super interesting. And I wanted to ask one question. It kind of relates to also what I'm researching now, and it's mostly how do you actually separate politics from ethics, and also how do you stop applying human value system and human politics onto microorganisms, and how do you position them? How do you see natural laws as actually something that exists out of human legislation and human society in a sense?
I'm going to answer your question with a question. You know, one of the great values of having artists in our community is that they're asking the hard questions. They're asking the new questions that we encounter as technology changes the way that culture functions. And so that exact question is perfectly reasonable art practice and something that should be explored. There are no easy answers to questions like that. Yeah, we're stuck in our box, we're stuck in our perspective, and it's always going to be an impossible challenge to try to see the world through the sensory organs of other creatures. It's something that, you know, philosophers have debated for a long time. But yeah, in some ways we're stuck in these bodies and stuck with our brains, and it's almost impossible to see it from other perspectives. So how do we do that? I mean, is that the role of art? You know, how? I don't know the answer. Elizabeth, can you give a more something?
I'll answer that question with a reading recommendation, actually, that you might be familiar with. But Robin Wall Kimmerer is an author who's a botanist and an indigenous woman. She belongs to the Potawatomi, and she has written a number of books, but Braiding Sweetgrass is one of my favorites. And in that book, she talks about developing reciprocal relationships with more than human organisms. So how, and this is a strong tenet of a lot of indigenous cultures, but very much so in Northern Native American populations, of how can you develop lifeways or maintain lifeways with reciprocal relationships with your environment and with other organisms in your environment. So how can you develop like ways of knowing and ways of seeing and ways of observation that allow you to determine whether that animal or plant is consenting to use by a human being, you know? So she talks about like, how can you tell that the plant is willing to be harvested for your use? How can you tell that the deer is willing to die for your use? And so she talks a lot about this being actually a very clear communication mechanism. So it's very clear to her when she can collect or harvest a particular plant and when it's not okay, or, you know, when that animal is willing to give up its life. And so I think that what's really interesting to me is how can you you know, translate these communication mechanisms to organisms that you can't see. So there's a way to think about, to observe plants and animals in kind of a direct and intuitive sense, especially when you know an environment very well. So you can tell, you know, like, I know that there are X number of, you know, sweetgrass bushes in this particular environment. And so I know that I can harvest this amount and I need to leave the rest of it for that ecosystem to reproduce. So those are all kind of intuitive means of observation that you can develop over time, but that's a lot trickier with organisms that you can't see. And so I guess what I invite you to do in response to this question is think about, you know, what are reciprocal, how can one define reciprocal relationships with organisms that you can't see?
I think that's such a fascinating question because it's so, you know, so I love Braiding Sweetgrass, but I also felt like she struggled with that question all the way through that book. And, you know, there were points with Sweetgrass where I think she might have found some peace, but there are other chapters where you can see with other organisms, she just she doesn't find that peace, that she's in some ways at war with other organisms. She talks about, you know, just like her, you know, doing landscaping around her house. You know, another good book is Staying with the Trouble, Donna Haraway, which is a goodie. But yeah, I mean, I think that, you know, because these organisms are never going to sign the contract with you, and you can make assumptions about what is good for the organism. And there's, you know, she has data that shows that some organisms do even better when you pick them, and then they grow even more under those quote unquote stress conditions. But, you know, does that make a happy, healthy organism? Does the organism accept that you're gonna, you know, pick its leaves? How could you possibly know? It's just sort of this impossible question. So yeah, I think it's a good one to think on. And I love that, like, you know, there may be a methodology that we can develop that we can actually make those readings. I think that's such a cool idea.
Yes, for sure. And also it's also interesting to think that the spore of life, the beginning of life, might have come also by that type of pushing the limitations of different organisms into what maybe at certain particular they were out of the possibility of each of them by themselves. No? So maybe I really hope that this dialogue can continue, and I'm sure that a lot of the residents will follow up, and hopefully you will hear from them and then projects at Biodesign Challenge. And I think that it's a beginning of a long conversation, I hope. And I really want to thank you both, Elizabeth and Daniel, for the fantastic lecture. And we certainly will keep in contact, and we will follow up into that. Thank you very much.
Thank you for having us.
Thanks for having
thank you
us.
for everyone for staying.
Fantastic.
Thank you so much.
See you soon.
See you soon. Thank you very much.